Hello guys! I'm Ladybird H. Suarez and welcome back to my YouTube channel. So for today's video, we are about to discuss the microbial structure. But before we will start, please don't forget to subscribe my YouTube channel and click the notification bell for more updates and information about microbiology.
I just need to clear my head.
Microbial structure. It is a gel-like matrix composed of water, enzymes, nutrients, waste, and gases that contain cell structures such as ribosomes, chromosomes, and plasmids. The cell envelope encases the cytoplasm and all its components. Unlike the eukaryotic or true cells, bacteria do not have a membrane-enclosed nucleus. Example is bacteria. It is classified as prokaryotes, which are single-celled organisms with a simple internal structure that lacks a nucleus and contains DNA that either floats freely in a twisted, thread-like mass called the nucleoid, or in a separate, circular pieces called plasmids. Structurally, there are three agricultural regions: the appendages, attachment to the cell surface in the form of flagella and pili or fimbriae. Second is the cell envelope. It is consisting a capsule, a cell wall, and a plasma membrane. And the third is the cytoplasmic region. It contains the cell chromosomes or the DNA and ribosomes and various of. Difference between microbial and bacterial. Bacterial is a subset of microbial called microbe. Generally just means anything small bacteria, yes, fungi, even protozoa. It usually does not include viruses. Bacteria is often used informally as a synonym for prokaryote, a term encompassing two of the three kingdoms of life, the eubacteria and the archaea. Many structural features are unique to bacteria and are not typically found among archaea and eukaryotes. Because of the simplicity of bacteria relative to larger organisms and the ease with which they can be manipulated experimentally, the cell structure of bacteria has been cell-studied, revealing many biochemical principles that have been subsequently applied to other organisms. What are the extracellular structures of bacteria? In bacteria, it consists of three major components. It is the polysaccharides, protein, and extracellular DNA. These components serve as key structural elements in multicellular assemblies called biofilms, in which cells stick to each other and are shielded by the ECM or the extracellular matrix. The extracellular matrix functions to communicate by sending messages from one unit to the next using physical connections called cell junctions. There are types of cell junctions are tight junctions, desimosomes, and gap junctions. Tight junctions. Still adjacent epithelial cells in a narrow bond just beneath their apical surface. They consist of a network of clothings and other proteins, perform two vital functions. They limit the passage of molecules and ions through the space between cells. Desimosomes are specialized adhesive protein complexes that localize to intercellular junctions and are responsible for maintaining the mechanical integrity of tissue. The last one is the gap junction. It is a specialized intracellular connection between a multitude of animal cell types. They directly connect to the cytoplasm of two cells, which allows various molecules, ions, and electrical impulses to directly passes through a regulated gate between cells. Prokaryotic cell structure. The parts are cytoplasm, nucleoid, capsule, cell wall, cytoplasmic membrane, ribosomes, pili, and flagella. The functions of prokaryotic cell structure. Cytoplasm. It provides a medium for the organelles to remain suspended. The cytoplasm acts a buffer and protects the genetic material of the cell and also the cellular organelles from damage caused due to movement and collision with other cells. Nucleoid. It is essential for controlling the activity of the cell and production. It is where transcription and replication of DNA takes place. Third is the capsule. It considered as virulence factor because it enhances the ability of bacteria to cause disease. It can protect cells from engulfment by eukaryotic cells. Next is the cell wall. The cell wall surrounds the plasma membrane of plant cells and provides tensile strength and protection against mechanical and osmotic stress. It also allows cells to develop turgor pressure, which is the pressure of the cell contents against the cell wall. Cytoplasmic membrane. It's also called as plasma membrane, is the most dynamic structure of a prokaryotic cell. Its main function is a selective permeability barrier that regulates the passage of substances into and out of the cell. Ribosomes have two main functions decoding the message and the formation of peptide bonds. These two activities reside in two larger ribonucleic protein fascicles or RNPs of unequal size, the ribosomal subunits. Pili are short, has-like structures on the cell surface of prokaryotic cell. They can have a role in movement but are more often involved in adherence to surfaces, which facilitates infection and is a key virulence characteristics. And the last one is the flagella. Are primarily used for cell movement and are found in prokaryotes as well as some eukaryotes. The prokaryotic flagellum spins, creating forwarded movement by a corkscrew-shaped filament. A prokaryote can have one or several flagella localized to one pot or spread out around the cell. The importance of microbial structure. Permeability barriers that allow selective passage of nutrients and inclusion of harmful substances. Adhesions used to attach or adhere to specific surfaces or tissues. Enzymes to mediate specific reactions on the cell surface important in the survival of the organisms. Protective structures against phagocytic engulfment or killing. Antigenic disguises to bypass activation of host immune defenses. Endotoxins, generally cell wall components that cause of inflammatory response in the host. However, the strength of the cell wall is responsible for keeping the cell from bursting when there are large differences in osmotic pressure between the cytoplasm and the environment. Cell wall composition varies widely among bacteria and is one of the most important factors in bacterial species analysis and differentiation. That's all for the microbial structure. If you like this video, please don't forget to like, comment, and share for more updates. That's all for the microbial diversity. If you like this video, please don't forget to like, comment, and share for more updates.